should be good with recording. So like I said, if at any stage you have questions, you just let me know and I'll pause the recording if you don't want to ask them on that. So this is just for the sake of recording, this is the Wednesday evening, 8 o'clock session. And this week we're really going through the material that relates to your exam. And you also have the lab practical to do, but there's no new material. And if you have joined me before to a session, I'm just saying this because this came up today, there's no requirement to join to all of the sessions. And also, do not feel that you have to be on a live session to get an attendance for the week. As long as you submit work, that counts as an attendance for the week. So there's nothing to worry like that. But of course, I always enjoy when you guys join me. So what I'm planning to do today, and what I was hoping that we would spend majority of our time, is to focus on the review for the exam. So I have a study guide for it, and we'll use that. But there's a couple of other things that I just want to mention, like I always do, that are kind of important things and so on. So I'm just going to jump out of this view, and we're going to go and look at the website. So we're going to go to the canvas, and we'll just go to my version of the canvas, provided that I find it. Here we are. Let me just get that down. Ooh, I didn't mean to share that. Hold on a second. We'll get the right screen up. This seems better. So you should be seeing the this course on the web browser. And what I'm going to do, I'm going to jump to the student view. And just so that I get everyone on board and anything that you might need for this session, I just want to highlight the study guide, provided that now we got to the student view. So when you go to this week's learning module, which is the week 4, you'll notice that there's under the lectures study resources, there's a study guide for exam 1. You're gonna come up. So if you click on that, too much love. If you click on that, there's gonna be a PDF file that you'll be able to access. And that's the study guide, and I have blanks here and there on that. And whenever there's a blank that's probably an important concept or an important term that I want you to be familiar with. You can also download the study guide by clicking the blue link. So if you want to use that while we're going through the material for the exam, I recommend that you either print it or download it or just open it on the side. However, I know that there's different learning styles and some of you might not find that ideal for yourself. So if that's not working for you, you just do whatever works for you. But that's how I've kind of planned it, that you can use that to guide you through everything that we cover on this week's session. So I'm going to jump back to the PowerPoint, provided I find it there, and you should be seeing again this week's objectives. So I just want to start real quickly. I don't know if it's more of me being excited than it is students, but I'm pretty psyched about the fact that we've made it to the week 4, because this is the first of the 4 exams that you'll take as part of the course. So by the time you finished this week, you have basically finished almost 1/4 of all the work that we'll be covering. So I think that that's super exciting. At least I always get excited when I kind of have an idea, where are we standing with the workload? So consider that it might have been a lot and a quick start, but by the end of this week, roughly 1/4 done. And for this exam, we'll be covering only 2 chapters. Whereas for the all other exams, it's typically 3 chapters that we cover. So also take it as a kind of on that view that even though it's your first exam and you're kind of still getting to know how I administer the exams and the entire format, at the same time, we try to make the workload a little lesser. So it's kind of a soft landing or soft start. But I do want to say that this class has been extremely good, and both of you, with submitting work. And if you ever have any issues with that you can't get work submitted on time, just let me know. I'm really chilled out about getting the extensions in place. So what I want to also remind you is that this week you don't have any new assignments. So there's nothing new that you need to do. However, you will be doing the exam and you'll be doing the practical. But when you've done the exam and you've done the practical, this week is ideal for catching up on any work that you might have missed. So I encourage you to let me know if you need anything reopened or if you need an extension for anything. The easiest way is to send me a message here on Canvas. But I also check the email on daily basis. But the big thing is that be very specific at what assignment you need opened. I sometimes get messages that are just, oh, I didn't get it done. Can you keep it open? And then I have to go by assignment to look it. I will still find it, but it might take a little longer before you hear back from me. So that's kind of all that I want to say. Not because I'm lazy to not do the work, but you'll hear back from me quicker. But my main goal is that you get all those points collected throughout this course, because really, like I said at the beginning, the way to succeed on these online classes is to collect every single point that you can get, and then at the end you can just chill when you already have a bunch of those in place. So like I said, this week what I'm asking you to do is do the exam. So we'll be revising the material for the exam now, tonight. And then you also have the lab practical to do. And both of these carry quite a bit of points. Exam 100 points. So if you compare it to what I showed earlier on about the points that you accumulate on a typical week. Last week we had 2 labs. But exams are worth of a good amount of points. So I really would encourage you not to skip those. It's better to at least guess some. They're multiple choice exams. So just get the likelihood that you get some points is high. You made it good. So that's for the exam. And for the lab practical, make sure to do that. That's about half of the length of it. And it's about half of the points as well. All of this work, I'm asking you to get it done by the end of the week. So on Sunday night, a minute before the midnight is when all of this is due, and the computer system will automatically close those exams and those practicals. However, if you have a valid reason why you can't get it done, you just let me know and I'll work with you again. But by default, it's easier for me to open coursework, assignments, homework than it is to open exams. So I just really encourage, try not to fall too far behind, because it is going to otherwise catch up, that then you're constantly doing catch-up game with staying on top of the workload. So I already mentioned about the study guide. So we will use that to guide our way through what's the important parts for this exam that you want to know. But before we jump there, I just want to highlight in terms of the lab practical. So lab practical is basically all the work that you've done before, and you've looked at all of that material when you've done those practice anatomy assignments on weekly basis. That's where you have those pictures and labels that you drag on right places, identifying structures, naming things, and so on and so on. And you remember, sometimes it's gross anatomy, things that we can see with naked eye, but sometimes it's also microscopy. So both are up for grabs. But basically, practical is all about anatomy identification exercises, whereas the exam covers then both anatomy and physiology. In terms of how to succeed with the practical, I really encourage you to work through those practice version for each time that we have an exam on that. That lab practical practice version includes questions that are pulled from a bigger pool. And if you play through that practice version a couple of times, you'll likely get all of those questions and you get to see them. The big difference is in terms of when you are actually taking the lab practical exam is that you don't have written options there that you then match. You actually just have a blank underneath, blank box, and you'll type the name of the structure that we're asking. In terms of should you be worried when you're typing the answers? No, even if you misspell something, you'll totally get the full marks. My spelling isn't perfect. If you don't know the answer to that, tell me something, tell me what it does, or tell me a nearby structure or something so I can give you partial points for that answer at least. So that's the secret with the lab practical. I'm trying to think if there's anything that I'm forgetting to mention of that, but I can't think of. And it's better to write something at least. And one thing that students often do, they get really worried once they've taken that lab practical and they get a score because the computer automatically wants to grade it. And it grades it in a really, really tough way that everything needs to be spelled exactly with the same capitalizations as whoever set up the practical has used. But that's not going to be your final grade. After everyone has taken the lab practical, I will actually go back to those and go through them question by question, student by student. And I think I go through them 3 times while being blinded to who the student is. And based on those 3, I'll then be able to make sure that everyone gets graded in the same way. And then you'll get your final grade. And I'll send an announcement of that when your final grade is up for grabs. Also, I received a question, I think last semester, few students were like, oh, it would really help if they would have from the chapters that we've covered in terms of the lecture videos and the kind of handouts. So the exam is going to be completely and only about the material that I covered on those voiceover PowerPoints that I've uploaded for you to use. So some students felt that it would be super helpful to have handouts of the slides that I use on those. I can't give the exact same handouts or exact same slides for copyright reasons. But what I've done, I've gone to your publisher's version of the textbook that you use, and I've done these 6 slides per page PDFs for you that covers the exact same material in slightly different words or slightly different arrangement, but it really is from one textbook to another, same stuff. You want to come up? No. So that's what I've got to say about the lecture slides. So what I'd like to do now is for us to jump and go to the study guide, and we'll work through that, and that's going to be the bulk of the session, like I said. But at this point, if you have any questions, just shout out. Otherwise, I'll just keep going and assume that everyone's happy. There will be time at the end to ask questions as well, but I really encourage that you shout out while we're going through material if something doesn't make sense. And the other thing is if I go too fast with the study guide. So let's jump out of this view, and I'm gonna open up the study guide for us to have a look of. So bear with me while I pull it up. Here we have it. So what you should be seeing should be a PDF reader of the first exam on BIO202 study guide. And like I said, we'll use that to work through all the material. I hope that this is good in terms of the size of the text, but if at any stage you feel like you can't see what's on the screen, you just let me know and I'll zoom in a little more. But what we're going to do, we're going to go through everything that you need to know for the exam. Every question in the exam is actually from this study guide. So there's nothing outside this. Remember that both the practical and the actual exam are open book exams. There's no proctoring. So you can use your notes. I would definitely make sure that I get the study guide completed and keep it close by. Remember, however, that they are timed. So I would be somewhat smart that I've seen the material before, that it's not the first time you start reading the textbook, because then you will probably run out of time. And that was the one thing that I was thinking that I needed to say about the practical as well. While you're doing the practice version of the practical, nothing prevents you from taking screenshots, taking pictures with your mobile phone, taking notes. Make it easy for yourself. All of those are allowed, even in that practical exam. So remember, I'm not trying to stress you out. I'm not trying to trick you out on anything. Make it comfortable for yourself. So we're going to start the first chapter that we had about the endocrine system, and how I normally try to start these, you'll notice very quickly, is that I try to somehow make a connection that how are the different parts of our course related to each other. And what we'll do, we'll start, like I said, with the endocrine system. And the big thing there that I think that we'll notice very quickly is that endocrine system is associated and deals with all other body systems. So really, the signals and messages that are sent from endocrine system affect everything else that we've got. But it's also a two-way track, that then different systems that we'll have a look of will have an effect on the endocrine system. So it really goes back and forth. So there's not really separation between the two. And that's the reason why when I said that this is probably the hardest chapter for most students, because really to understand fully the hormonal system, how hormones are made, how they work, how they are deactivated, and how they are removed from the body, you really need to know almost the other system as well. But because we can't just skip the endocrine system and then at the end dump it on you, we're going to do it at this point. So just don't feel that if this doesn't feel easy, that's not your fault. That's the fault of the layout, how the course is laid out. So you've seen before nervous system. I think you finished your BIO201 by talking about the nervous system. And we saw that nervous system was the one of the two body control systems, or if you wish, the messaging systems. And what we see there is that there's quite a big difference between how action potentials in nervous system and how hormones in endocrine system work. So whereas the action potentials are really fast and they last only for a very short amount of time, hormones, they take a longer time to act. Sometimes it can be minutes, sometimes it can be hours, sometimes it can be days or even weeks that they take before they act. And their effects are much more longer lasting. So they stick around in your body. Hey, take it easy. No need to be. Okay, we're gonna take a quick break. Hey, take it easy. We're gonna be able to continue. I think that the cat and dog had a chance to meet each other. So I think we're good. So we're gonna have a look of what are the big messages that you want to know about the endocrine system. Of course, the field of studying hormones, how hormones are made, how they act, how they are deactivated, how they are removed. That field is called endocrinology. So just so you know, again, in terms of the science, we have specific, hey, you take it easy. We have specific terminology that we use, and this is one of those. When you kind of want to have a look, what functions does the endocrine system serve? Where do we have hormones playing role? Well, it is pretty much in all parts of the body, all processes within the body. The big message that I want you to take away, you're going to take a chill pill now. Big message that I want you to take away from this is that all of that happens through the blood, through the circulatory system. So that's really the big take-home message. We've also before, I think on your chapter about the integumentary system, so of the skin and nails and hair, all that, we probably have talked about different kinds of glands. So what you might remember is that there's two kinds of glands in the body. And the first group of glands that we have seen were these glands that had, let's say we have the gland here, and they produce something. Let's say that this is a sweat gland. A lot of sweat glands are in the layers of the skin. They produce the sweat, and the sweat is released to the surface of the skin through a duct. And they act immediately at that site where the secretions are released. That's how I'm just listening, are the cat and dog fighting, but it seems that everyone's fine. So that's how these exocrine glands work. So good examples of the exocrine glands include sweat glands. Also think of the tear gland. So the lacrimal gland here and how it opens, you can see that little dot close to the corner of the eye on the lower edge. Salivary glands. We'll talk about 3 types of salivary glands later on this course. All of these have the gland and then a duct, and the duct opens into a very particular place. So that's how they work. But our interest is going to be on this chapter, the endocrine glands. So, of course, the hormones. So these endocrine glands, they differ significantly that we have the gland here, it produces something, but all of these secretions are released to the bloodstream. And within the bloodstream, these secretions travel to all parts of the body. They're not specific where they travel. And then when the hormones reach the part of the body where we want it to act, that's where they will then trigger the response. However, remember that hormones are going to travel throughout the body. So that's why I always say that messing with the hormonal system through pharmacology is one thing that I avoid doing because there's just so much that can go wrong. So big messages there, bloodstream transporting the hormones and act throughout the body. And we'll go through all of the major endocrine glands during this session. And then knowing us instructors, we always love something when it's a little odd or when it's a little off. So this is the case here as well. So there is one gland in the body that's both endocrine and exocrine. And that's going to be pancreas. And we'll talk about pancreas later on. And we're not really going to be that interested about the exocrine function. Exocrine function of the pancreas is going to be the production of the pancreatic juice, which is the most complete digestive juice that your body releases. So that's what breaks down the food. You get the food into your body, but then it needs to be broken down small enough that it can pass through the plasma membrane from inside your digestive tract to the inside the rest of the body. So we'll talk all about that when we look at the digestive system, but on this chapter, we're of course focusing on the endocrine function of the pancreas. And that's going to be the regulation of the blood sugar levels. So there's going to be 2 important hormones, insulin and glucagon, that you're going to have a look of, and we'll talk about those once we get to the pancreas. So you have probably seen, I'm just laughing because I see the dog looking for the cat. You probably have seen this handout. This is the handout we completed during our very first session when we were going through the material of this chapter. And this handout is going to be super useful because it really includes the key points, the key glands, and what they produce, what hormones, and how those hormones act. I'm not going to spend the time to go through that on this video or this session. So if you've missed any of that, you have the revision video from the week 2. So I encourage you to use that to guide you if you feel lost with any of that. We're going to go through those same glands, but we're going to go through them in a little different format. And that's what we're going to do the next. And I think what we're going to start with is going to be by looking at the hypothalamus. So you might remember when you covered in the BIO201, the different parts of the brain. Just make yourself comfortable. We covered the different parts of the brain. Hypothalamus was the part roughly in the middle of the brain. When you look, you separate the two hemispheres and then cut through. That was located right underneath the thalamus. And when we look at the hypothalamus, there's from it a little, I'm just going to redraw that. From the hypothalamus, there's a little stalk that comes out. And at the end of that stalk, we will see our pituitary gland. And pituitary gland is actually going to have two different lobes that we'll look at. But we're now focusing at the beginning on this, the hypothalamus here. And often the hypothalamus is described as the master gland of the body. And that's because it is the hypothalamus that really starts these processes of how hormones are released. So it really controls pretty much all other glands, or many other glands at least. And how hypothalamus receives them signals, that can be in two different ways. And these signals that it receives, they come from the other areas of the brain. So they can be either neural impulses that pass as action potentials to the hypothalamus, or they can be hormones that travel to the hypothalamus and then trigger the action there. Well, now that we've had a look of the hypothalamus, I want us to get a name for this stalk that I mentioned. So the part that connects the hypothalamus to the pituitary gland, right here. This is going to be known as infundibulum. So that's the anatomical name when we have a structure that connects one structure to another. And you'll notice that we'll see this term elsewhere as well. But in this context, that refers to the part that connects the hypothalamus to the pituitary. So what we're going to do next, we're going to focus our attention on the pituitary gland. And we're going to go through that in two different parts. And if we consider that this individual who we drew there, they would be looking to the right on the screen. So of course, this side is going to be the posterior lobe, and this is going to be the anterior lobe. Ooh, I didn't mean to jump ahead. So we're going to start with this posterior lobe and look what happens there. So posterior lobe receives signals from the hypothalamus in two different formats. It receives these signals both as a neural signal, so action potentials, but also as a hormonal signals, so the endocrine signals. Just like we talked about the hypothalamus. Same is true for the pituitary gland's posterior lobe. So let's add that to our diagram. And if at any stage you feel that you didn't get something and I'm moving too fast, you can just give me a shout out. I normally do come back, so don't worry. I'm not planning to skip anything so you can't see it. So the message has now passed from the hypothalamus to the posterior lobe of the pituitary. And there's going to be two hormones that we need to know. So the first one of those is going to be the oxytocin, and second one is going to be antidiuretic hormone. Be careful, it's her toy. So the dog has stolen cat's toy, so that's the drama going on. So let's start with the oxytocin. So oxytocin is a hormone that plays a role in the childbirth. So during the delivery of the baby at the end of the pregnancy. Another time when we see oxytocin levels rise and when it affects is when the milk is produced and released from the mammary gland. So both of those are actions of the oxytocin. Oxytocin is a weird one because that's going to be one of the very few times in the body when we actually see positive feedback loop of the homeostasis. And what we'll do, we'll go through every step of the homeostasis in just a moment. But at this point, just remember that that's an unusual example of seeing that positive feedback loop that we see with oxytocin. The reason why it's so unusual is that usually when we have homeostatic mechanism, the feedback is going to be negative, and that means that it tries to stop something from happening. So we don't want whatever is happening to continue happening. But in positive feedback, what happens instead is that something happens, and this feedback mechanism makes it happen even more. So in terms of giving birth to the child, when the child starts to come out and the uterine wall lining is pulled or pushed, we're not going to stop that halfway through, leaving the baby hanging out there. No, that stimulation is going to trigger the rest of the muscles of the uterus and lower parts of the abdomen to push down, squeeze the baby out. Same goes for the lactation as well. When the baby starts suckling the nipple, it doesn't stop us from making the milk. It actually promotes making and releasing the milk. And more you make and release milk, more the baby will suck the nipple. So it's kind of an add-in to that effect. So that was our first hormone that we needed to look at from the posterior pituitary. The second hormone that I want to show to you is going to be ADH. And ADH stands for antidiuretic hormone. Hold on a second. Oh, take it easy. We're gonna continue in a second. Hey buddy, take it easy. You can't be so excited. No. You don't need to be that excited. Hey, you're gonna hang out with me. Sorry about that. The dog is just going to hang out with me now. So, hey, take it easy. You're just going to sit down with me. So antidiuretic hormone does exactly what it says in the tin. So it is all about urination. And when we have anti, it's against urination. So we are producing less urine whenever antidiuretic hormone is released. So there's times, say when you're dehydrated, for example. Well, in response to being dehydrated, you will start releasing or producing antidiuretic hormone that gets released from the posterior lobe of the pituitary, and that makes you make less pee. We want to preserve all of that, all of those fluids in the body. So that's all you need to know about the posterior pituitary. What we're going to do, we're going to now move on and have a look of the anterior pituitary. And anterior pituitary actually receives all of its messages from the posterior pituitary. So anterior pituitary doesn't receive messages directly from the hypothalamus. These messages first go to the posterior and then are passed to the anterior lobe of the pituitary gland. And then those hormones are secreted from there onwards. So if we want to complete our diagram here, oh, I didn't mean to do that. We can add the color here. We're going to be focusing now on this, on the anterior lobe of the pituitary. hey buddy, you need to take it easy if you can't behave. And there's going to be 5 hormones or groups of hormones that I want us to focus on. The first one that we're going to look at is going to be the growth hormone. Well, growth hormone is often abbreviated as GH, and growth hormone does exactly what you would expect it to do based on their name. It regulates the growth of an individual. So when we see growth hormone levels being high, it's going to be, for example, right after the baby has been born. The development and growth of the baby is very fast during the first year or so. Another time when we see a lot of growth hormone and that kind of spiking up is during the puberty. So during the puberty, we grow a lot in size, and we also keep developing much further. Your lab is way too intense. So that's another time when we see a surge in the growth hormone levels. Well, the second hormone that I want to talk about from the anterior pituitary is going to be thyroid stimulating hormone, often abbreviated as TSH. And again, it does exactly what you would expect it to do. It makes the thyroid gland work more actively. So it promotes the action of the thyroid gland. I think the cat went to the front room. You're just going to hang out. Honestly, make yourself comfortable. Yeah, he's good. So we're going to talk about the thyroid gland in a second. So it is the thyroid stimulating hormone that makes the thyroid gland work more active. So the third hormone that I want to discuss from the anterior pituitary is going to be actually adrenal gland hormone. And again, what adrenal gland hormone does, it promotes the adrenal gland to do its job. So you'll notice that this is why we think that hypothalamus and pituitary are kind of these master glands or control glands. They control how other endocrine glands work. Well, we're going to talk about adrenal gland promoting adrenal gland hormone promoting adrenal gland in a little bit of a moment and have a look what adrenaline does. But before that, I want to mention 2 more groups of hormones. So the fourth one, which is actually 2 hormones, a set of 2 hormones from the anterior pituitary, are going to be FSH. Ooh, take it easy, don't upset the kitty. Are going to be FSH and LH. The FSH stands for follicle stimulating hormone. So this hormone promotes the development of what's to become the egg cell. Hold on. Hey. You take it easy. You don't need to bark to the cat. You're just going to be stuck with me now. So we're going to talk about the development of the egg cell and all that process as the very last chapter of this course. But at this point, I just want to flag it up on your map in your mind that the egg cell, which is the follicle, the early egg cell, the development of that is driven by the follicle stimulating hormone from the anterior pituitary gland. Another hormone that affects the female menstrual cycle is going to be the luteinizing hormone. Luteinizing hormone is actually a hormone that triggers for that egg to be released from the ovary. So we'll talk about that in a great detail as well. Hey, buddy, you are behaving so naughty that you're stuck with me. You're going to have to take part on lecture. And the last hormone from the anterior pituitary that we're going to have a look of is going to be the prolactin. So prolactin does exactly what you would think it does based on the name. So lactin part of the name refers to the lactation, and pro, that it promotes lactation. So it's the combination of oxytocin and prolactin that results in the milk production and milk release in a female who has given birth. So those are our hormones that we needed to know from the pituitary gland. At this point, because whenever I jump off to the screen, I lose the chat. I'm just checking. I don't see any questions, but if you have any questions, just shout out. And we're going to try to work with this rebel dog. So what we're going to do next, we're going to move on and we're going to be looking at the adrenal gland. And I'm not so sure if you've heard about adrenal gland before. Certainly if you have not done a lot of AMP, it might be one of these structures that's a little bit stranger to you. So where we find adrenal gland is on top of the kidneys. So if we look at a kidney is kind of this bean-shaped structure, of course bigger than a bean, and it has a tube coming out from there. The right kidney is slightly lower because there's a liver mainly on top of it, left a little higher up, and the adrenal gland is located on top of these. On both sides, we've got one adrenal. And adrenal glands are a little bit, if you wish to describe them, like a pyramid-shaped structures on top there. And we're going to be looking at the adrenal gland in a way that we look at two different distinct parts of an adrenal gland. So what I'm drawing here is a cross-section of an adrenal gland, and you'll notice that the outermost part is, of course, going to be our cortex. So we've seen cortex already before when we talked about the brain. You remember that we noticed that the brain had this sausage-looking structure outside the outermost layer. It was also darker, so gray matter. Well, that was the cortex of the brain. So just like that, we also have cortex of the adrenal gland. So we'll have a look at that. And after that, we'll talk about the medulla of the adrenal gland. So medulla is going to be the middle part of this gland. No, you're just stuck with me because you can't behave. So you'll see the medulla here. So let's start with the cortex first. And there's actually going to be 25 different hormones that we find being secreted from the cortex of the adrenal gland. I'm only asking you to know two groups. So they're groups because I've bundled up a bunch of them together. And the first one that we're going to start with is going to be our aldosterone. So aldosterone acts on the kidneys. So it's not a surprise that in addition, it would have an effect on the urine production. And the main function of the kidneys is actually regulation of the blood pressure. So that's not going to be surprising either. Blood is made of plasma, like we'll see on the second chapter that we're going to review. So it's not surprising that it affects also the concentration of that and also the energy production as a process of making ATP. The reason why I wanted to highlight the aldosterone is that there is a medical condition called aldosteronism where too much aldosterone is being produced. So if you want to think what would be the effects of having too much aldosterone, well, it would be all these things going hyper. The kidney would work super fast. Urination would be running on overboard, blood pressure regulation, we would be hyper of that, energy production, and so on and so on. So that would be a problem. The second hormone from the cortex of the adrenal gland that I wanted to mention is actually a group of hormones, and these are our glucocorticoids. So if you might have heard about hormones such as cortisol or hydrocortisone, cortisone on its own, cortisol, corticosterone. There's a bunch of different ones that fall underneath that. And these hormones are really the hormones that are produced in response to stress. So any one of you who has severe allergies or skin issues, you might end up getting cortisone locally on your skin as an ointment treatment. In certain other medical conditions, we can inject cortisone into, for example, a knee joint if there's damage there that can help with that, and so on and so on. So really the effects of the glucocorticoids is to help to deal with damage. You secrete glucocorticoids in response to the stress, and it doesn't just have to be psychological stress. It can also be physiological stress, like in terms of injured knee. But let's say that we think of students during the exam week. Pretty much all of them will have elevated glucocorticoid levels. That's because they're running on that stress response. That's what's keeping them going. So after the exam week, many students, for example, come down with a cold. That would be an example of you having been just keeping going on because of the stress and stress responses. Well, let's have a look at the medulla of the adrenal gland. And two hormones that I want to highlight from there are going to be the epinephrine and norepinephrine. So those are two important hormones for us to know, and these play a role in increasing your heart rate, for example, affecting how your blood is distributed in the body. So they play a role also in that. Really, overall, all of these sympathetic nervous system responses. So you remember when we talked about the autonomic nervous system, you had two branches. You had sympathetic and parasympathetic branch. And the sympathetic branch was this fight or flight responses. So when you are either fighting for your survival or you're doing the best you can to run away from that situation. Well, we noticed that epinephrine and norepinephrine are secreted when this branch of the autonomic nervous system is activated. So in this case, your heart rate goes up, your blood pressure goes up. We direct the blood not to your digestive system or urinary system, but we direct it to the muscles, skeletal muscles, so you can run away from that situation or you can fight against that situation. Things like that. The other branch was, of course, the parasympathetic nervous system, which was the rest and digest response. So from this, I want to highlight the hormone adrenaline, kind of going to what we talked about, the epinephrine and nephrine. Are you going to be good? If you get into a fight, you're going to be stuck with me. You can go, but only if you're going to be good. So in addition to epinephrine and norepinephrine, I'm trying to go down here with this arrow. We're going to look at the adrenaline, and that's going to be the third hormone to highlight there. So adrenaline, of course, drives the sympathetic nervous system branch. Be good. So that's all I want to say about the adrenal gland and the cortex and medulla of the adrenal gland. I hope that makes sense. The next one that we're going to look at is going to be the gonads. And gonads are the primary sex organs. So for the females, let's get a nice pink for that. It's going to be, of course, ovaries. So the female ovaries don't get into a fight. The hormones that are going to be secreted from there are going to be the estrogen and progesterone. So often when I ask from students, what's the function of the ovary? They say that, of course, the production of the egg cell. That is true. That's what ovaries do. But ovaries, just as importantly, do actually produce the female sex hormones. And these are the aforementioned estrogen and progesterone. And we'll talk about the estrogen and progesterone in much more detail when we look at the female and male reproductive system as the two last chapters of this course. So that was for the females. Let's have a look of the males. Male primary sex organ is, of course, going to be the testes. So what do testes do? They do produce sperm, but in addition to that, they produce, especially the important hormone that we're going to look at is going to be testosterone. And again, the effects of the testosterone, we can talk about those in a greater detail later on, but we do produce the sex hormones during the early embryonic development in terms of ending up with one or another sex, and then later on life as well. But both of these hormones are present early on. Next one that we'll talk about is gonna be the kidneys. And I think that I already drew the kidneys there, so I hope that I'm using roughly same color. Let's jump and have a look. Yep. So we use this orangey brown for the kidneys. So let's have a look of kidneys as a hormonal gland. What do we need to know about that? Well, whenever we talk about kidneys or kidney sciences, you don't go and see a kidney doctor. Actually, you go and see a renal doctor. You don't go and get an X-ray or CT image or MRI of your kidney. You go and get a renal imaging done. So the term renal gives us a rise to this hormone that's going to be the important one from the kidneys. And this hormone is going to be renin. So what renin does, it plays important role in blood pressure regulation. So you remember the main function of the kidneys was to regulate your blood pressure. So that's through the hormone renin. We're going to come back to leptin, so don't freak out. I'm going to come back to it and you're going to have a chance to write all the notes about leptin. But I actually want to move on and have a look of the pineal gland first. And pineal gland is a gland that we find deep within the brain. If you had a chance to look at that video of the sheep brain dissection that I uploaded on the week 2 material, I'm not going to test you on that video. That's not something that you need to know. It's just something that some students were very interested in. And I said, okay, since we cannot dissect in this class, since we're online, I can't really put sheep brains into an envelope and send them to you. At least you can see a video. So I went and recorded a video. And when you look, the pineal gland is about P-shaped structure located by the third ventricle of the brain. So between what we know as the little brain and big brain. So roughly there. And what pineal gland does, it secretes a hormone called melatonin. And be certain that that's not melanin. Melanin is something totally different. That's what gives you the skin color. Melatonin is a hormone that plays a role in regulating your sleep-wake cycle. So, for example, individuals who suffer from sleeping issues, they might take melatonin. Or people who travel from multiple time zones to another, they might take supplementary melatonin to help them to fall asleep. So melatonin is something that your body produces yourself, but you might have to rely on that if you have issues with that. The next one, the pancreas. We already touched upon the pancreas earlier on. I mentioned that was the odd one. That was the one where we had both endocrine and exocrine function. Well, this time, because we are focusing on the endocrine functions, we're looking at the hormones. The two hormones that I want you to know from the pancreas are going to be the insulin and glucagon. So what they do, they help to regulate the blood sugar levels. So if you want to, and we'll have a look at the pancreas in a greater detail later on this course, but if you want to kind of roughly imagine where it is, it's within the abdominal cavity. We would have our stomach here, and this is not to the scale, esophagus coming down, and the first part of small intestines right after the stomach, which is going to be called duodenum. It's wrapped up into that kind of arch. Let me put on a charger so we don't run out of battery. So we should be good. I don't think that we're gonna... Now we should be good. I don't think that we're gonna run out of battery. So reason why we're gonna talk about the pancreas and the blood sugar regulation is, of course, the clinical condition of the diabetes mellitus. So diabetes type 1 is something that you're born with. In that, for example, your body would not be making enough, or it would not be receptive to making enough insulin. So these patients who have diabetes type 1, they end up having to take insulin injections for pretty much all of their life. And that's just something that's going to be the case. There's really no way around it. Type 2 diabetes is actually late onset diabetes. So that's something that you develop later in life, and it can be managed fairly well with the changes of the lifestyle. So that's the type 2. Be sure to know the difference between the two. And again, we're going to come to parathyroid gland in a little bit. Before that, I want to discuss about thyroid gland first. Hey! Take it easy, buddy. Do you need to come to my lab again? Are you going to come to my lab again? I think he is. He's over here now first. You might be taking to the bedrooms. Are you coming with me? You're going to study with me. Yeah. Big boy. Sorry about that. I tell you, dogs and cats, they take a while to learn to like each other. So we're going to have a look of the thyroid gland. So I don't know if you've heard about the thyroid gland a lot in the past, but it's located at the neck on the anterior side, just inferior to the larynx, and often it's described in the textbooks as being a butterfly shaped. And I do think that anatomists are sometimes very creative of seeing these structures and shapes in various parts of the body where I don't necessarily see those. But the butterfly shape refers to the fact that thyroid gland has two lobes that are connected by this thin bridge. And the thin bridge is called isthmus. So that's our thyroid gland here on the neck, just roughly where you have your Adam's apple on top of there. What thyroid gland does, it produces a hormone, and this hormone is going to be T3. T3 hormone actually gets converted into T4 hormone, and the big thing here is that these hormones play a role in metabolism, regulating your metabolism. So often when we have individuals who have either extreme weight gain or weight loss, and we cannot find another reason for it, not in terms of the lifestyle, not in terms of what they eat, we end up noticing a lot of the time that that's because it can be that their thyroid gland isn't working exactly as it should be. So you can have overactive or underactive thyroid gland. Let's look at the situation where your thyroid gland is overactive. In that situation, you could eat all you wish and you're still not going to be able to put on any weight. Your love is too intense. You're not able to put on weight. You just have a very fast metabolism. You burn off that energy. Your BMR, basal metabolic rate, is really, really fast. Well, how about, what can you do about that? Well, we can take medicines that interfere with this conversion of T3 to T4. The other thing that we can do, we can actually cut away part of the thyroid gland. I'm an anatomist, so I always like to cut into people. That's what we would do. If your thyroid gland is underactive, the best thing we can do is give additional T3 or T4 hormones. So there's a couple of clinical examples that I provided there as well. And the reason why I wanted to talk about the thyroid gland before the other glands that we're having a look, we're going to go backwards, is that the next gland that we're going to look at, I didn't mean to do that. Next gland that we're going to look at is actually going to be parathyroid gland. And parathyroid gland are these little yellowish dots on the thyroid gland. So they're tiny, tiny, tiny glands within on the surface of the thyroid gland. So let's have a look of those. So we're going to talk about the parathyroid glands. This looks better. So they're these tiny yellowish, yellow-brown glands on the thyroid gland. But if you don't take it easy, I can't let you go. You've been so naughty. So we're going to have a look of the parathyroid gland. And for the sake of consistency, the color looks good now. So what parathyroid glands do, not probably surprisingly to you, they produce parathyroid hormone. And why parathyroid hormone is important and why I want us to look at it is its role in the calcium homeostasis. You might be wondering that why do I care about calcium? Well, calcium is actually involved in pretty much everywhere in the body. So we'll see a bunch of examples of reactions where calcium takes place role as we go through the material on this course. So there's a lot to do with that. Some of the examples I've listed there, urine production, kidneys, intestines, digestion, not surprisingly skeletal system, and also vitamin D activation. But just know at this point that these parathyroid glands, they're located on the thyroid gland and play a role in calcium homeostasis. Well, that brings us to talk about the thymus. So let's have a look of the thymus a little bit. Buddy, you're gonna just go and cause trouble, so you're stuck with me. That's how I get my friends. Someone causes trouble and they have to hang out with me. So let's look at the thymus. Thymus is a kind of again, I'm saying this as an anatomist, it's kind of a tricky one because we do see thymus on a newborn and on children, but older you get, your thymus actually disappears. What happens? It shrinks and it gets replaced by the connective tissue and adipose tissue. So on an adult and especially on an older person, I would not be able to find thymus. Thymus is located just above the heart on the anterior surface of the chest cavity. So that's where you would look for it. But the reason why I want to talk about thymus with you is that when we look at the immune system, it is to the thymus where the T cells, a special type of white blood cells, go and mature at. And even though your thymus shrinks and is not as visible when you grow older, it doesn't mean that you wouldn't have T cells anymore. It does still have its physiological function. It's just not going to be as significantly visible. Now, the last hormone that we have left is going to be the leptin. And the reason why I left leptin as the last one is that it's hard to pinpoint it to a specific body part. And that's because it is actually the fat tissue, the adipose tissue, that produces leptin. And you might wonder, why would this be the case? Well, the case is that leptin is a hormone that helps us to control our appetite. So whether you're hungry or not. So we sometimes have situations where an individual, especially a younger individual, is just morbidly obese, and we can't really understand why they don't have that natural stop for eating. They just keep eating and eating and eating. Well, the explanation to that might be in the leptin hormone. So we've done some experiments, and I was lucky enough to be part of a research group that worked with mice, lab mice, from who we actually removed all of the leptin receptors. So they just kept eating and eating and eating, and they did not have any ending to their desire of eating. Not just to the point that they were full, but they ate so much that their stomach, for example, could give up. They physically ate themselves to death. So what we've seen with some of the patients and human patients who have had issues with uncontrollable weight gain, and there's no other explanation, once they've started to get additional leptin injections, actually they have returned to the normal body size and normal match with their developmental stage. So that's all the hormones, that's everything we covered. in our review session on the week 2, there is a little bit more to this chapter, and we're going to go through that because that more that's to this chapter is still lecture content, so it might still come up in your exam. And I think that the best thing to start, since we've been speaking about hormones, would be that can we give a definition to hormones? I think defining things sets our thinking always straight. Don't get into trouble. So hormones are chemicals, and these chemicals travel in the blood, so in the cardiovascular system, to all parts of the body. Another place where we see hormones is sometimes in the lymph, but we're gonna just leave that as on the back of your mind, just that it can travel there, but the blood is the main site. And there's 2 types of chemicals that we're gonna be having a look. We're going to be having a look of the autocrines and paracrines. Well, autocrines are these chemicals that are secreted. What I'm drawing here is supposed to be a cell. So we have a nucleus and cell membrane. Autocrines are these chemicals that are secreted by this cell and act on the same cell. They don't even have to leave the cell itself. Paracrines, instead, are secreted from one cell, and they travel not too far away to the same area, but they act on another cell or other cells. So it's not the same cell where we're seeing the response to paracrines. So those would be the 2 types of chemicals, and I'd like you to be comfortable with those so you kind of know what's the difference between the 2. Then in terms of the chemical structure of the hormones, this is more of just like good to know. We are not going to go into great detail. Some hormones are made of amino acids, derivatives of amino acids. Some are made of steroids, and steroids are actually made of the cholesterol. So you do need to have some amount of cholesterol in your body. However, all the cholesterol that you need is produced by your body, so you don't need to eat cholesterol-rich foods. And then there's the 3rd group, eicosanoids, and we don't really know, should we classify them as hormones or paracrines? So we're not quite certain of that, and we're still trying to figure out that. So as you can see, science is not, we don't have all the answers always. It's not fully with the answers. We're still doing research. Well, the next question that I ask from my students normally in face-to-face classes is that, okay, if hormones are released from one gland and they travel to all parts of the body, why don't they then change the behavior of all the cells in the body? Wouldn't that make sense? That's, I think, a very good question. And really the secret or the answer to that is going to be within the receptors. What receptors are, again, I'm drawing a cell here and a nucleus. Receptors are located on the surface of a cell, and it is hormones that tie into these receptors, and then when this bind between the 2 happens, that triggers some kind of a change in the cell. It can be activation of the gene in the cell, or it can be a production of protein or some other metabolic processes. But there's only receptors on the cells where that particular hormone should act on. So we've got many different kinds of cells, we've got many different kinds of receptors, but not all receptors and all hormones are a match. It's almost like it would be this, and this is a really bad drawing, I already know it, so apologies. It's almost like it's this key and a lock mechanism, that the 2 need to be a match. And it needs to be a perfect match. So they're very, very specific. And like I said, what they do when a hormone binds into a receptor, somehow it alters that cell's activity. So it triggers some sort of a chain of events inside that cell. And I've listed many different examples of what it could be. Well, the next thing that I want to mention, and this might seem a little vague initially, that why do I care? But think of not just the facts that it happens, think of the consequences of that. Some hormones are water soluble and some hormones are lipid soluble. And I think that the best way to see the difference is, again, let's think of, we've got two cells here. One cell here and another cell here. And first ones that we're going to have a look of is water-soluble hormones. Well, these water-soluble hormones, there's no way that they can pass through the plasma membrane. Remember, plasma membrane was made of phospholipid bilayer. So there's lipids there, and water cannot cross a lipid layer. So the way how water-soluble hormones act is that they have to bind into this receptor on the surface of the cell, so on its plasma membrane. And then a second messenger, so something else has to travel inside the cell as a result of this receptor activation. So it's not the hormone that makes its way inside the cell. Well, this is the case with the water-soluble hormones. Situation is different with the lipid-soluble hormones. Well, now they can actually go through the plasma membrane. They can go through this phospholipid bilayer. And there they can trigger different kinds of events inside the cell. But they don't need this second messenger. They can trigger the changes directly within the cell on their own. So that's the big difference between the two. And I've listed here a bunch of information. I think I took this from the lecture notes that I had on the lecture video about the similarities and differences between water and lipid-soluble hormones. So that's just there for good to know in case you're interested. And now we get to the concept that I warned you about before, and it's not warned because it's horrible or it's boring. The reason why we are going through this is actually something that I think is rather a topic that we should have covered earlier on your A&P studies, but because I know that I haven't been teaching you, we're going to cover it now. And the reason why we're going to take time to cover it is because it is such an important concept. So we're going to talk about homeostasis. And homeostasis was, of course, the process of maintaining the environment inside the cell so that it's consistent, so there's no changes, or maintaining the environment not just inside the cell, but inside the human body. So we are maintaining a consistent internal environment within the body. And that's what the homeostasis means. And you see many different kinds of diagrams in many different kinds of textbooks. There's seesaw kind of diagrams, many arrows everywhere. I've condensed everything you need to know about homeostasis into 4 boxes. So we're just going to need to know 4 steps. So remember, body is always looking for a change of some kind. And this change is what we consider as the stimulus, something that triggers for this homeostatic mechanism loop to be initiated. And this change is detected by a sensor of some kind. And these sensors can be many different kinds. But what sensors do is that they're constantly screening. Is there a change from a normal? And if there is a change, then they send a message to the brain. And brain is typically the control center in the humans and many other animals as well. So the message gets passed to the control center. The sensor itself doesn't do thinking. It just simply detects something and triggers an action potential to the brain. Well, within the brain, we give a meaning to this change, to this stimulus that we've seen. And within the brain, we often see a set point. And the set point is something that's the range of acceptable normal values that we see. So let's take temperature for an example. So we can have very cold temperatures and we can have very hot temperatures. Your body temperature is roughly 37 Celsius degrees. If you go to negative 35, negative 34, you start to get sick. If you go to much colder, you're going to die. If you go to 40 Celsius degrees positive, actually your enzymes will start denaturating. So your enzymes will start to get destroyed, and as a result, you won't be able to function. So we need to keep this body temperature within a very narrow range. And you can think of this as the set point. Set point is almost like if you have an AC unit on the house and that box in the wall where you adjust how cold or how hot it can get. That's the set point. Well, if we deviate from this set point to either direction, the brain tells that we need to act. So that's some sort of a effect, some sort of a response to this change. And usually, most of the time in the body, the response that we're going to do is going to be negative response. So what we want to do, sorry, I lost my writing tools. What we want to do is to stop that change from happening. So in engineering, this line that's blocked by a line like this, that means that it stops the stimulus from happening. So let's have a look at our example of temperatures. We notice that you're getting too cold. That's not good. You're going to die if you get too cold. You get sensed that by thermoreceptors, which are all over your body. Your brain says that, oh, we're getting cold. We need to do something. We don't want to freeze to death. Well, you can have many different kinds of effectors, many different kinds of actions in response. You can start shivering, in which case your muscles produce heat. You can start directing blood to the vital organs. So let's maintain our brain, lungs, heart. Those are things that we can't live without. You can have behavioral responses, not just physiological. You can move to a warm environment. You can put on clothing. You can curl up. Things like that. So those stop the stimulus from happening. These sort of responses are known as negative feedback because they stop that stimulus from happening. And most of the responses that you see in the body are gonna be negative feedback. There's only very few times when we see positive feedback. And we already talked about few examples of that. So in positive feedback, that stimulus is detected, passes a message to the brain, and then that triggers us to do more whatever causes that stimulus. So it amplifies that stimulus. So examples of that, it promotes that happening would be childbirth. I talked about the baby's head pushing against the uterine wall lining. That stretches the uterine wall lining, which triggers the mechanoreceptors that feel a stretch on that to pass a message to the brain in the set point there, what size the uterus should be. And rather than trying to stop that stretching from happening, now we actually promote that. We start pushing from higher up on the uterus to push the baby out. So childbirth is a great example of positive feedback. The other one that we talked about was lactation. When the baby suckles the nipple, that causes... Oh yes, of course, go for it. I will upload the video. It's going to be available there, the rest of the session. Go for it. So when the baby suckles the nipple, that will cause milk to be released. And more milk you release, more baby suckles that nipple. So it's again like amplifying that response. One more that we haven't talked yet is, thank you, have a good night, is going to be blood clotting. So if you cut yourself, you start bleeding and you will form a scab. A little scab produces more scab to be produced. So that promotes itself. Those were all examples of positive feedback. One last thing that I want to mention about the endocrine system is that it doesn't work separately from the nervous system. These often interact. So hormones can trigger some sort of a nervous system response, some sort of an action potential, for example. And the other way around, an action potential that we saw sent from the other parts of the brain to our hypothalamus triggered then hormones to be released. So there's a lot of overlap between the two. So don't let yourself kind of be led think that because we're talking of these as separate, that they wouldn't be connected. So that's where we got there. And let me see if there's anything else important that we should say. The next one is probably pretty self-explanatory. If you have some sort of a response to a hormone, well, that response depends on three things, that what kind of a response we get. It depends how much of that hormone we have in the blood, and also how much receptors we have for that hormone. So those two would affect how much we get of a response, and also how tight of a bond is formed between the hormone and the receptor. So those three things determine how much activation we get within a cell, a target cell, based on a hormone. Sometimes we might have a situation where we don't produce a lot of hormone, and that hormone is still important for us. So we want to be aware of when there is that hormone. And if we don't produce a lot of hormone, what we do in response to that, well, we actually make more receptors. This would be known as the upregulation. So if we don't have a lot of hormone, we still want to detect that hormone, why don't we just make a lot of receptors then? If even there's a little hormone, these receptors will catch it. The opposite of that is when we have a lot of hormone around, and we don't want to go crazy that, okay, now there's a lot of hormone around, so we're going to get a lot of response for it. Instead, let's balance off the situation and remove some of the receptors, so we're not getting as strong of a response to that hormone. This would be known as downregulation. So be sure to know the difference between those two. Those would be a good ones. I think that would make a nice question in an exam. And some hormones are free in the blood, some are bound, but really the concentration of the hormone depended on how much of it is released, how much of it is inactivated, and how much of it is removed. And there's many different places or locations where hormones get removed by kidney, by liver, and by certain enzymes especially. And I think that the last thing about the endocrine system that I want to talk about are these three kinds of interactions that we see between different hormones. And there is a question, if I remember correctly, where you will be matching the description and the term of these. So that's why I want to go through those. I know we've spent a lot of time on hormones, but I think it's going to be worthwhile. So if we have one hormone here that normally would do some kind of a response, but that hormone requires another hormone to be present so that it can work, that is known as permissiveness. So you get a permission to work by another hormone. Let's look at the next case. In this case, we have this initial hormone, and the other hormone being present makes this initial hormone act even stronger than it would normally act. So it's synergism. So it amplifies the effect of that hormone that we had. And the final case that we can see is going to be antagonism. If any of you reads a lot of books, you know in every good story, or watching movies, in every good story, there's an antagonist. So that's someone who's like opposite or against of the star of the story. That's the bad guy. Well, what happens here is that we have this initial hormone that would go and do its job. But because there's a second hormone, it blocks this initial hormone from doing its job. So it acts against of the initial hormone. So that would be antagonism. So I would know these definitions for these three kinds of interactions, so you would be able to match them with the terms. Again, remember, it's an open book exam. As long as you've taken these notes, you're set. You've got all the information you need. Okay, that concludes our notes from the endocrine system. And if you don't have any questions, I'm just gonna jump right on to the chapter on the blood. But I know that it's been a lot, so if you do need to go, I totally get it. I totally understand. And I will just finish off this and then you can watch it online if that's something that works better. But just putting it out there that I understand if you have to go, but I'm super happy if you stay as well. So it's up to you. Let's have a look on the chapter about blood. And this is going to be quicker. This is going to be much easier. I think that endocrine system, because it is so vague, it's easy to get really carried away with it. But what I want to do at the beginning, like I've done before, I want to kind of set the scene. On this chapter, we're looking at the blood. And blood is really the liquid within the cardiovascular system, so the transport media. And then on the future chapters after the exam, we'll focus on the heart, which is the pump that drives this liquid inside this cardiovascular system. And we'll also have a look at the tubing of the cardiovascular system, which is of course going to be the blood vessels. So this is how all of these 3 chapters will be connected. But for our exam, you just want to know endocrine system and blood. So we're not going to jump too far. And as we're getting started, I think that a good way or good thing to consider is that we have 4 main types of tissues in the body. Well, which of those would blood be? Well, blood is connective tissue, and this sometimes throws students off because we think of connective tissues as tendons, ligaments, bones, things that connect structurally, physically different one part to another. Well, it can be also just a physiological connection. So blood, even though it's liquid, it carries stuff from one part to another part. So it's connecting those parts. So just remember that that would make a good question. What kind of a tissue is blood? It is a connective tissue since it connects different body parts. And if you want to describe it somehow, you can describe it as the internal transport system. So it really is this transport media that we saw earlier on within the body. And a lot of things are transported in the blood. Well, blood is made in the bone marrow, actually in the red bone marrow. And here's an important term. We'll keep naming these processes of making different kind of blood multiple times during this session. So when we are talking about general blood, not one particular component of the blood, but blood in general, and what is the making process of the blood called? That's called hematopoiesis. So poiesis means making of something, and hemato is the term for blood. So if we're talking about generally making blood, that's going to be hematopoiesis. And if you wonder how much blood you have in the body, it's about 8% of your body weight. So that's on an average individual, roughly 5 liters. Well, why do we need to know about the blood? Well, really drawing blood from a patient and then analyzing that blood sample is one of the most common and one of the also easiest and most simple clinical tests that we can do in medicine. And I read some time ago a study, which I think the number is probably pretty accurate. About 70% of the clinical decisions that the medical doctor or a nurse makes is based on the results of the blood work. So that's why it's so important that you know the basics of the blood as you continue your career into nursing or other health careers, medical school, medical imaging, nursing assistant, dentistry, wherever you're going. So let's have a look at the functions of the blood. And there's a bunch that we can associate with the blood. Probably if I would have to pick one of these, the most important function that I would highlight of this is the transport function of the blood. So blood helps us to transport, move materials from one place to another. And for example, our cells in various parts of the body need oxygen. And they also need nutrients so that these cells can work wherever in the body they are. And the way we get that oxygen from the lungs to those cells, or from our abdomen where we get the energy and nutrients from the food to wherever the cell is located, is by transporting it in the blood. Similarly, we need to get rid of waste products that are produced as a result of cellular metabolism, and also carbon dioxide that's produced in that process. Well, we take those away from those cells and transport them either to the lungs to breathe out the carbon dioxide, or to the kidneys to get rid of most of those wastes. And of course, we've already talked about the hormones on the previous chapter, that hormones as well are transported within the blood. Some other functions that I've listed there, yes, blood plays an important role in regulation. So blood is mainly fluid. So to move fluids from one part of the body to another, let's move blood. Also, water finds a lot of the heat. So it's easy to move heat from one part of the body to another by moving it with the blood. And same goes also for the pH value. So we maintain our pH value. There's a homeostatic mechanism loop that maintains it within very narrow range. And we're able to maintain it within very narrow range because even if there might be local differences, the blood circulation quickly normalizes those. And then we, of course, have the protective functions of the blood. We're going to talk a lot about immune system in a couple of chapters. And, of course, the blood clotting. We're protecting us from bleeding to death. So those are some good examples of the functions of the blood. Like I said, I would especially highlight this transport function of the blood. Then let's have a look at the composition of the blood. And this is really all that there is going to be for this chapter about the blood. So how do we analyze blood in the most simple way when we don't have microscopes and we don't go crazy? Well, what we do, we take that blood sample, draw it from a patient, and then we put it to a centrifuge, which spins that test tube where the blood is like crazy. And while it's spinning, the different components of the blood will weigh a different amount. And based on their weight, and I'm trying to draw here a test tube in case you're wondering what is that supposed to be. Well, based on their weight, when you put a sample to a centrifuge, different elements of the blood move to different height or different location on this test tube. The lightest elements go to the top of the test tube, and the heaviest elements go to the bottom of the test tube. So let's have a look of what elements will we then end up seeing in there as a result of this spinning the sample. Okay, and the first one that we're going to look at is going to be the plasma. So plasma is all this liquid within which all other parts of the blood are dissolved in. So plasma is the only liquidy stuff. Everything else is either cells or parts of the cells. And there's going to be about 55% of the blood is going to be plasma. So it's going to be the lightest, so it's going to be at the top of the test tube here. About 55%. And if you've ever seen plasma, it's this kind of a yellowish, or I've described it here as a straw-colored sticky fluid. It's a little stickier than your water would be. Why plasma might be useful in clinical practice? You don't need to do donor-recipient matching. You can just simply give a transfusion of plasma. Everyone can take everyone's plasma. If you remove all other parts of the blood from that blood sample, you can give plasma to anyone. Well, we also find that a lot of proteins such as albumin, globulin, and fibrinogens are dissolved in the plasma. And then there's a bunch of other things we see: nutrients, gases, hormones, waste products, inorganic ions. So all of those that we see being transported in other ways as well. So that's our plasma, and that's all you need to know about plasma. All other parts of the blood that we will be discussing are gonna be actually concrete things. They're gonna be either cells or parts of a cell. And these are known as formed elements. So rest of the blood sample is gonna be formed elements. So let's go through those. And there's gonna be 3 different formed elements in the blood. And the first one that we're gonna look at is gonna be red blood cells. And after plasma, red blood cells are actually the most numerous part of the blood. So about 45% of the blood or volume of the blood is going to be red blood cells. And we often abbreviate it as RBCs. So let's add our red blood cells to this diagram of ours. And red blood cells are heavy, so they're going to be at the bottom of this test tube. So red blood cells about 45% here. And the term that I want you to know is here, the erythrocytes, but just because it's such an important term, I'm just highlighting it here as well. So erythrocytes are the scientific name for red blood cells. And the reason why I wanted to make a big deal about this term erythrocytes was because when we talk about production of the red blood cells, we end up with what we just saw us talking about earlier on. So the term poiesis referred to the making of something. Well, in this case, when we are making of red blood cells, it's going to be erythropoiesis. So we're using this particular term that applies to the red blood cells. So be sure that you can tell what's the term for normal or just blood production in general, which was hemopoiesis. What is the term for production of the red blood cells, which is going to be the erythropoiesis. And we're going to talk about that also for the white blood cells. Well, what does a red blood cell then look like? We describe them often as being biconcave discs. That might be a difficult description, so let's just draw it out here. So red blood cells are kind of these flattened discs, if you wish, and they're squished from the middle. So you could almost think of them like donuts that haven't been cut all the way through. So they're like balloons that you squeeze from the middle. This term biconcave disc comes from, if we take a cross-section of this red blood cell that we just drew here, let's cut it in half here along this plane. What we see the red blood cell looking like is gonna be, oh, I lost my tools there. Put them back. That worked out fine. So the cross-section of a single red blood cell is gonna look like this. It's thicker from the edges and squashed from the middle. So that's what a red blood cell looks like, but not fully cut through from the middle. The other thing, that's what the word biconcaved means, that it's concaved, caved in from the middle on both sides, so bi. The other term that we sometimes hear when we talk about describing red blood cells, what they are anucleate. The anucleate means that there is no nucleus in them. In fact, there's no other cell organelles either. For example, mitochondria in the red blood cells. And if you think that that sounds a little weird, all cells had a nucleus. How did we end up with this thing that now all of a sudden red blood cells don't have a nucleus? Well, red blood cells do have a nucleus initially, but they lose it during their development. And they also lose those organelles within the cell. And that makes sense if you think of the function of the red blood cell. The function of the red blood cell is to transport things, nutrients, but also especially oxygen. If we had all these cell organelles, the red blood cell would end up consuming all of this oxygen. And at the end, when it has done its travel, there'd be nothing to give to the tissues. So that's why we don't have cell organelles in the red blood cell. So that was a crash course to the appearance of the red blood cell. We're also going to talk about what it looks like in terms of the color. And when we talk about its color, let's draw a red blood cell here again. I keep losing my tools. I'm so sorry about that. So let's draw a quick diagram. This is a red blood cell. Round cell. And within every red blood cell, one red blood cell can fit in four hemoglobin subunits. There's 4 of them located there. And these are often abbreviated as HB, capital H, lowercase b. So these are hemoglobin that we just drew there. And notice that there's 4 of them. Well, what hemoglobin does and why it's so important for us in a discussion of the red blood cells is that every hemoglobin can carry 1 oxygen atom. So a single red blood cell can carry in total 4 oxygens. And we had 4 hemoglobins. So sometimes we can look at the blood, and especially the red blood cells, based on at how much oxygen does it have. Well, if we have a lot of oxygen in the red blood cell, so it's rich in oxygen, we talk about being oxyhemoglobin. This sort of a blood, and especially the red blood cells, are bright red in color. So they are that blood that when you cut an artery, out flies this bright red blood. If you watch any movies where they do fighting and they end up cutting themselves, that would be oxyhemoglobin blood. However, sometimes some of that blood is going to be low in the oxygen concentration. Say, for example, if that blood is returning back from tissues, that's going to be called deoxyhemoglobin. So oxy for oxygen and D, that there's not a lot of oxygen. This sort of blood, it's not really blue, although in often textbooks you see it being drawn as blue, but it's more of a purplish, darkish blood. So now you know where those 2 different colors come. And now, why did we need to talk about hemoglobin? Well, there is a clinical relevance to that as well that I want to highlight. So sometimes you might have certain gases such as carbon monoxide. Carbon monoxide is produced when something doesn't burn completely. Also, cigarette smoke contains carbon monoxide. Well, we all know that you can die out of carbon monoxide poisoning, so that's not great. What happens in this case is that the carbon monoxide binds to the hemoglobin subunit, and once it has bound to the hemoglobin subunit, oxygen is unable to bind to this hemoglobin. So it takes the place of the oxygen. So this is why if there's a lot of carbon monoxide in the air, one will die because your body is not able to receive enough oxygen because your red blood cells, hemoglobin units, have already been taken by this carbon monoxide. Same goes for smoking. I keep telling you smoking is bad for you. Okay, a couple of more things about the red blood cells that I want to highlight. We want to aim to a situation where the amount of red blood cells that we make is equal to the amount of red blood cells that get destroyed. So we want to have them two in balance. But sometimes that's not the case. Sometimes that doesn't happen. If you have too little red blood cells, we talk about you having hypoxia. If you have too many red blood cells, the blood really becomes quite viscose, so easy to slide past each other. How do we control the amount of red blood cells in your body? Well, it's going to be a hormone, and this hormone is called EPO. That stands for erythropoietin. And since we've been talking about hormones on the previous chapter, I think that this is a good one to know. What erythropoietin does, it makes your body to produce more red blood cells. So where we see people using EPO is in athletes a lot of the time. So if you go to a high altitude, what happens in high altitude is that there's very little oxygen there. Well, in response, your body starts producing EPO. And when you produce a lot of EPO hormone, your body starts to make a lot of red blood cells. So these individuals, these athletes that have all of a sudden more red blood cells than their competing athletes, they have, in theory, an advantage over their competition. Because if you're able to carry a lot of oxygen to the muscles, these muscles can work much harder. So that's actually allowed. Many athletes go before important competition to train in high altitude training camps. What is not allowed and what is considered as doing doping is if you just take EPO hormone and inject it into yourself. That's cheating. So you take what you want out of that story. You can also affect the amount of red blood cells by your diet. For example, your diet does need to include certain amount of iron, amino acids, lipids, and carbohydrates. Well, red blood cells live in your body somewhere between 100 and 120 days. And once that time comes up, we break the red blood cells into its components, to a heme, iron, and globin, and these can be reused, so recycled. And the last thing that I want to say about red blood cells is when we have little oxygen in blood, as we saw, the response to that was the EPO. I just try to make space that I can write there. Well, if you have little oxygen in the blood, that's known as being anemic. So some of the signs and symptoms of anemia include being pale, tired, having breathing issues, being constantly cold, and so on. That can be due to many different causes. Maybe you're losing blood, so internal bleeding can make you anemic. You can produce too little red blood cells, or you might destroy too much red blood cells. So there's all these three factors that can play a role why one becomes anemic. But there's a specific type of anemia that I want us to discuss about. And the reason why I want us to discuss about it is that that is gonna be your extra credit question in the exam. So what that means that in the exam, you've got, I believe, 50 questions and 60 minutes to answer those. So about 1 minute per question. Well, in addition, you have one extra question. The 51st question is up for grabs in terms of getting extra points. That's a short answer, so mini essay question. And I tell you ahead of the time, this time that what it is, it's all about sickle cell anemia. I'm going to ask you, what is sickle cell anemia? What causes it? What consequences does it have to have sickle cell anemia? So why is it a bad thing to have sickle cell anemia? And why could it be a good thing? And then I ask you what it makes you think about evolution. So let's go through all of those. So we saw earlier on that a normal red blood cell was this flattened disc that had a depression in the middle, right? Well, this is what gets coded in a DNA. So just the amino acid chain that gets produced as a result of this DNA code makes this nice flattened disc shape for the red blood cell. However, sometimes what happens is that we have errors in the DNA. The error can be just one single amino acid, or one single base pair. So one single, if you think horizontal line of that step ladder that has been twisted around, if you want to describe DNA like that. And that one single base pair difference causes one amino acid difference in the resulting red blood cell. And as a result, that red blood cell's shape is going to be all wrong. It looks like this crescent moon shaped cell. So it's not what we're used to seeing. It has sharp edges and it is only kind of thinner shaped curved like that. Well, this is not great. The reason why it's not great is that there's two things that result out of this. First of all, these sickled cells cannot, so it's sad thing, they cannot transport as much oxygen. Transport less oxygen. Other thing that happens is that they end up getting caught and causing blockages, thin, small, very narrow blood vessels. So both of those things would be a problem with these sickled red blood cells. So not a great thing, you might say. Well, if we look who does have typically sickle cell disease, we find out that individuals who are either from sub-Saharan Africa or whose ancestors came from sub-Saharan Africa have typically sickle cell disease. They are more likely to have sickle cell disease than any other group in the world. So you might think that that kind of sucks for them, right? They're a little bit unlucky. Well, turns out they're not really unlucky. They might be luckier than we are. And you ask, why would they be lucky? Because they have less oxygen being transported and those red blood cells might get caught and blocked. Well, if you don't exercise like crazy and if you kind of are able to manage how much oxygen you need, you might be just fine. And you might not even get those blockages if you're kind of lucky and not pushing your body to the extreme. It turns out that there is another thing that's very common in sub-Saharan Africa. And the other thing that's very common in sub-Saharan Africa is malaria. So malaria is this parasite that enters into your red blood cell. Ooh, I thought I had a pen on. Let's see. Ooh. The malaria parasite enters to your red blood cell as part of its life cycle. There it multiplies in numbers until there's loads of malaria parasites and they leave the red blood cell, and as they are leaving the red blood cell, they might burst the cell and kill the cell. So that's not great. Getting a malaria is not fun. Well, turns out that malaria parasite is not able to enter sickled red blood cell. So it turns out that people from sub-Saharan Africa are less likely to get a malaria. And if you have ever had a malaria, you know that that's not fun. And also, if you have had to take anti-malaria medication to protect you from possibly getting malaria, you also know that they are rough medication. Many people lose their hair as a result of anti-malaria medication. So maybe it wasn't that individuals from sub-Saharan Africa who have a sickle cell anemia are unlucky. Maybe it is that evolution has came up with a clever way of protecting them against something that's common only on that area. And we did not realize before that it's actually beneficial. You don't have to believe in evolution, but since this is a science class, we work on an assumption that evolution happens. And this would be a good example of evolution over generation, coming up with something that's actually protective. So it would be the individuals who have sickle cell anemia that would be more likely to survive in sub-Saharan Africa. So that's all you need to know for your short answer question. You can write it ahead of the time on a notepad or Word or wherever, and then just copy paste it there. I'm expecting about one paragraph, so maybe 4 or 5 sentences. You don't have to go crazy. Just a simple explanation. What is sickle cell anemia? What causes it? Why this might be bad? And then consider why it could be good. Well, the malaria is the thing that we're discussing here. And then what does that tell you about evolution? So I'm giving you away the answer to the short answer question. That's all you need to know about red blood cells. And we only have 2 more things to discuss before we wrap up. We're going to talk next about white blood cells. And white blood cells are often abbreviated as WBC because we're just too lazy to write them out. And the scientific term for white blood cells is leukocytes. And again, this will be important later on. And there's about less than 1% of the blood volume is white blood cells. Let's go back to this diagram that we drew about the test tube. And the white blood cells are in their weight somewhere between the plasma, and red blood cells. So they're kind of in a weight in between of those two. So they end up building up in between those two layers. Less than 1% of the total volume of the blood. Well, what white blood cells do, they protect you against various different kinds of diseases. So we'll talk about the immune system in a great detail later on this course. But at this point, all you need to know that white blood cells are part of the immune system and they protect you against diseases. And there's going to be 5 types of white blood cells that I'm asking you to know. And in an exam, you do not need to be able to identify the white blood cells. But you will need to be able to know when you are shown the names of the white blood cells, then what's most common and what's least common and everything in between. However, in the practical, you do need to be able to identify the different kinds of white blood cells. So I'm leaving it to you to go through what do they look like. I have very good lecture videos that I provided to you, and also the practice anatomy covers that really well, that what do different kinds of white blood cells look like. So let's go through the facts that we need to know about white blood cells. The most common white blood cell type that we have is going to be neutrophils. Somewhere between 50 to 70% of all the white blood cells in your body are going to be neutrophils. They have a multilobed nucleus, so it looks almost like a pearls on a string, staining very dark when you stain them. The cytoplasm of the white blood cell neutrophil is going to be full of these little dots, granules. What neutrophils do, they protect you against various different kinds of immune issues. So they have a protective role in that. I'm just thinking, is there anything else that I was supposed to say about white blood cells? I can't think of. I think that's about it. Just remember, immune defense is their function. Let's look at the second most common white blood cell, which is gonna be our lymphocytes. There's somewhere between 25 to 45% of the white blood cells in your body are lymphocytes. They have a large spherical nucleus and no granules in the cytoplasm. So we describe these, if there's no granules, they're agranulocytes. There's only a little rim, pale part of the cytoplasm visible. Rest of the lymphocyte is gonna be that nucleus. What lymphocytes do, they produce antibodies and they're involved with the immune responses to protecting you. And there's 2 types of lymphocytes, T cells and B cells, that we're gonna talk later on this course. But the big thing to remember, the T cells mature in the thymus, B cells mature in the bone marrow. So that's all you need to know about the lymphocyte. The third most common white blood cell type is gonna be our monocytes. Somewhere between 3 and 8% of all the white blood cells. Their nucleus is more of a kidney shaped, so you saw that kind of a bean shape. There is no granules in the cytoplasm, and there's a lot of pale blue area showing up. What our monocytes do, they're actually phagocytic. So this process of phagocytosis refers to eating pathogens that shouldn't be in your body. So these monocytes travel around in the body. When they find something that shouldn't be there, they just engulf it and eat it. And then they break whatever is inside them over time and get rid of the broken particles. So that's the way how they protect you. So phagocytosis is the name for that process. The fourth most common, or the second least common white blood cell type that I want you to know about is going to be eosinophils. Eosinophils make about 2 to 4% of all the white blood cells in the body. They are binucleate, so they have two nucleus, two lobules of nucleus. It is still one nucleus, but it's made of two lobules, which stains very, very dark. The cytoplasm has granules. And what they do, they actually act against parasites. And they play an important role in allergies as well. So that's eosinophils. And the least common white blood cell type that we see is going to be our basophils. Somewhere between 0.5 to 1% of all the white blood cells are basophils. They have two lobules of the nucleus that stains very dark, granules in the cytoplasm. And what they do, they actually play a role in inflammation responses, and also allergies. So when you take antihistamines against your allergies, you're actually controlling basophils. That allergy is a natural protection mechanism gone out of the scale. It's gone crazy. So basophils are attacking things that aren't even a problem. So antihistamines keep those in place. You stop those basophils from going crazy. So those were our 5 types of white blood cells that we needed to have. Our neutrophils, our lymphocytes, our monocytes, our eosinophils, and our basophils. You'll notice that I listed them in order of most common and least common. I can tell you that I am going to ask in an exam for you to organize the different white blood cells based on how common they are, from the most common to least common. And you will be given those different white blood cell types, but you have to organize them. So for that reason, I wrote the first letter of each one there. And we're going to go to the next page to see why I wrote that down. If you use that order of the first letters, you'll see that it makes sentence, never let monkeys eat bananas. We love mnemonics in the science, especially in the anatomy and physiology. So that tells you the number, how common. These different kinds of white blood cells are. And the production of the white blood cells, well, that is gonna be, again, a certain kind of poiesis. We remember that poiesis referred to the making of, production of something. And because we're talking about leukocytes, it's gonna be leukopoiesis. So production of white blood cells was known as leukopoiesis. So that's another type of production process of cell type that you need to know. And just a couple of clinical things that I want to mention about white blood cells. If you have too much white blood cells, so overproduction of them, that's known as leukemia. Too little white blood cells is known as leukopenia. And we only have one more type of cells that make up the blood to discuss. I think that the discussion takes 4 lines, if I'm not totally mistaken. 4 or not a lot more. I promise that. So the last type of cells that we find in the blood are going to be platelets. And we're going to wrap up this review session with the platelets. I'll say a couple of words about the blood type matching, but we're going to talk about platelets as the last bigger of a concept. And platelets are also less than 1% in the amount of the volume of the blood. So it's also located here between the plasma and red blood cells. In fact, this layer made of white blood cells and platelets here is known as buffy coat, because it's kind of a coating there and it looks a little buffy. So let's see what we need to know about platelets. Platelets are not full cells. They're actually just simply fragments of cells. And what platelets do, they help with clotting of the blood. So when you cut yourself and bleed, it is thanks to this clotting mechanism that you stop bleeding. You don't bleed to death. Some individuals, including myself, don't make platelets like they should, so I need to take supplementary platelets. Otherwise, if I cut myself, I just keep bleeding, and will bleed to death. So that's why I'm lucky that pharmaceuticals are existing and take care of us, that we can give artificial platelets. Platelets survive in the body only for about 10 days, so I'm constantly taking them because they will only stick around for about 10 days. And that's the last part of the components of the blood. The very last thing that I will mention to you about this review guide is a little bit about the blood donor-recipient matching. So when you do blood transfusions, why is it important and why do we need to know about it? But we'll keep it very, very light. So you cannot give just random person to another random person's blood. That's because there will be otherwise a rejection that happens. So we're always interested about these donor-recipient matches. We want to make sure that they're a good match. Whoever donates the blood and whoever receives the blood. If they're not good a match, what happens is that the blood will actually end up clumping. And that clumped blood will cause issues. It will not transport oxygen and it will stick around and it can cause blockages. So let's have a look of different blood types that we can have. We can have individuals who are blood type A. They can receive blood from another blood type A individual and also from a blood type O individual. And I've discussed why this is in your lecture video. They cannot receive blood from a B or AB individual. Individual who's blood type B, they can receive blood from a blood type B individual and also from another O blood type individual. They cannot receive from a blood type A or AB. And again, I discuss why this is the case, why do we get this clotting in the lecture video if you haven't had a chance to see this. Individuals who are blood type AB, they can receive blood from anyone at all. They'll be fine regardless. And finally, individuals who are blood type O, they can only receive blood from other blood type O individuals. So this table tells us something that's going to be really important for us to take away from this story. It tells us that any individual whose blood type AB can receive blood from anyone at all. So these individuals, AB type, are known as universal recipient because they can receive blood from anyone at all. And individuals who are blood type O, they can donate their blood to anyone at all. So anyone can receive blood from a donor whose blood type is O. And finally, sometimes you hear about individual being A, B, AB, or O, either positive or negative. Well, this positive and negative refers to whether that individual has Rh factor or not. So Rh factors play a role and they're important if you have a mother and the mother is expecting a child that's still in the womb, and their Rh factors are different. One's positive, one's negative. That's not a problem during this first pregnancy. But what can happen, and I've never seen a delivery of a child, someone giving birth, where the blood from the mother and the child wouldn't have mixed between each other. That can happen, but I've never seen it happening. So if there's a mixing of these bloods and they're different Rh type, both the mother and child will be fine. But if that mother gets pregnant for the second time, there is a risk that that mother's blood will attack that baby that's still developing. So that's why the Rh factor is important, and that's why it's especially important in the second pregnancy. So that's all you need to know about the blood and all you need to know for this exam. And on that note, we're going to jump back to the PowerPoint just to wrap up this session. And what we see in this PowerPoint, I just want to remind you, if you feel that you would benefit from having tutoring, we do offer tutoring. You've got this link everywhere around. The Cranium Cafe is the virtual tutoring service that we offer. You do not need to have an appointment. You just show up there, and if there's a tutor that you especially like, you can even pick a particular tutor. And if they're free, you'll just be hooked up with them. If they're not free, you get put into a lobby, and then when they become free, they will see with you. The other option is that, of course, I'm available for you as well. I'm not formally a tutor, but I will, of course, make the time for you. So you can contact me anytime, and I'll make myself available for you if you feel that you want to have a one-on-one session, or if you feel that you want to organize a group, for example, your study group. I'm happy to facilitate those, or even as a whole group. So just let me know if I can help you. And that really concludes everything that I had for this session. I know it's been a long one. I appreciate you joining me. At this point, the only thing that I have left is to ask if you have any questions. If you don't, I think that we covered everything that we set out to cover, but I'm happy to answer any questions. If not, I thank you for joining me, and I will definitely make a note, Mackenzie, that you joined tonight and stuck around the entire time. It's usually in the final grade when I'm kind of figuring out if you're on the edge between two grades. I do look at who's been around, even though there's no requirement to be around. But if you've been around, I consider that as a positive thing. If you don't have any questions, this is it for tonight. If you have questions, just shout out or write to the chat. I assume that, okay, that's everything. Well, I really appreciate you, and I hope you have a good night as well. And thank you for sticking around. Don't stress over the exam. You've got every answer to every question in that exam. Have a good night. And that concludes our recording as well for this final review session that we had for this week. I hope you have a good week. Let me know if you've run into any issues with the exam, but I think you are all set for success. I'll see you next week.